In 2006, a scientist proved that a living adult cell could be erased. Not killed, not replaced, rewound, back to a state it hadn't been in since before you were born. Most people have never heard the full implication of that discovery, because the institutions that fund aging research are the same ones that profit from treating its consequences. By the end of this video, you will understand a biological mechanism that most physicians have never been trained to consider, and why the gap between that mechanism and clinical practice is not an accident. But the most disturbing part is not what the discovery showed. It is what happened to it afterward. In 2006, Shinya Yamanaka at Kyoto University published a paper that stopped the scientific world. His team took fully mature adult cells that had been a specific type for years, and exposed them to 4 proteins. Those 4 proteins, now called Yamanaka factors, erased the cell's identity entirely. What remained was a pluripotent stem cell, a cell that had forgotten what it was and could become anything. The Nobel Committee awarded Yamanaka the prize in 2012. 6 years. The fastest recognition in modern Nobel history for a biology discovery. But the finding that followed, published by David Sinclair's lab at Harvard in 2023, is the one that changes the question entirely. Sinclair's team did not reprogram cells to zero. They reprogrammed them partially. They restored function. They reversed measurable biological age markers in eye tissue. The mice recovered vision they had lost. Right now, inside your body, every cell carries an epigenetic clock, a chemical record of how much time has passed. That clock can, under the right conditions, be reset. But the mechanism behind that reset is something medicine has not been designed to measure. And the reason why is not what you would expect. Every cell in the human body carries 2 layers of information. The first is the DNA sequence itself, the genetic code. The second is the epigenome, a set of chemical tags that sit on top of the DNA and tell the cell which genes to read and which to silence. As cells age, those tags drift. Genes that should be silent become active. Genes that should be active go quiet. The cell begins misreading its own instructions. This is not damage in the conventional sense. The DNA is not broken. The cell has simply accumulated errors in interpretation, and those errors compound across decades, across every organ system, at different rates. What Yamanaka's factors do is strip those errors away. They reach into the epigenome and dissolve the accumulated chemical noise of years. The cell does not die. It does not divide. It is simply made to forget that time has passed. The cell does not know how old you are. It only knows what it has been told. And those instructions can be rewritten. In Sinclair's 2023 mouse study, partial reprogramming, using only 3 of the 4 Yamanaka factors, restored vision in animals with simulated glaucoma and age-related decline. The cells in the optic nerve were not replaced. They were restored. Function returned within weeks. And here is where the story becomes structural, not biological. The technology to partially reprogram human cells exists in research form. What does not exist is the pathway to bring it to a patient. And the reason for that gap is not science. The FDA's drug approval framework was built in an era when aging was not classified as a disease. It still is not. Because aging is not an approved disease indication, no pharmaceutical company can run a clinical trial with reversing biological age as the primary endpoint. The regulatory category does not exist. This is the structure. A regulatory system designed around acute conditions is being applied to a chronic, measurable, multi-system process, and calling it unapproved because it has not yet been defined. The result is that reprogramming research lives almost entirely in private biotech. Companies like Altos Labs, funded by billions in venture capital, running trials that will not reach patients for a decade or more. Your doctor has no instrument to measure your epigenetic age. Not because the test does not exist, it does, but because it is not covered, not standardized, and not part of any annual physical. Biological age clocks, Horvath's DNA clock, the grim age predictor, can estimate cellular age from a blood sample with measurable accuracy. They have existed in research form since 2013. They are not part of routine care anywhere in the world. A 2023 review in Nature Aging found that epigenetic age acceleration, the gap between biological and chronological age, predicts all-cause mortality better than most standard clinical markers, better than cholesterol, better than blood pressure alone. The test that could tell you how fast you are aging costs less than a standard lipid panel. It is not covered. It is not ordered. It is not discussed. But what the research shows can actually be done without waiting for regulatory approval is not nothing. And it is not what the wellness industry is selling. 3 interventions have demonstrated measurable effects on epigenetic age markers in controlled human trials, not animal models, human trials. The first, sustained aerobic exercise. A 2023 study in Aging Cell tracked epigenetic clocks in sedentary adults who began a structured aerobic program. Biological age markers reversed by an average of 3.24 years over 8 months. The mechanism is mitochondrial. Exercise signals cells to run repair pathways that are otherwise dormant. The second, caloric restriction with adequate protein. The Calorie Trial, a 2-year controlled study of 25% caloric restriction in healthy adults, found significant deceleration in epigenetic aging. The mechanism involves mTOR pathway suppression. The cell shifts from growth mode into maintenance mode. The third, sleep architecture. Deep, slow-wave sleep is the primary window during which cellular repair proteins are expressed. A 2022 study in Nature Communications found that even partial disruption of slow-wave sleep accelerated epigenetic aging by measurable increments within days. Your biology is not fixed. The epigenome is responsive, but it responds to signal, and most of what modern life delivers is noise. The window is not infinite. Epigenetic drift accelerates with age. The cells that can be reset today may not respond the same way in 10 years. In 2006, the question was whether a cell could be rewound. That question was answered. The question science is now asking is different. How far back can a living system go before the reset becomes something else entirely? Partial reprogramming without full dedifferentiation, the controlled version Sinclair's lab demonstrated, has been replicated in multiple tissue types: muscle, kidney, optic nerve. The pattern holds. What has not been replicated is the clinical will to measure it in living patients. Your chronological age is a number. Your biological age is a state. And unlike a number, a state can change. The science established that in 2006. The system has spent the years since deciding what to do with it. It has not decided yet.